Okay, so the next step is to share my screen. So greetings everyone. You have joined the event being hosted by the Santa Clara Valley chapter of IEEE Computational Intelligence Society and a number of other IEEE chapters in the country. My name is Vishnu Pendiala. I'm the chapter chair. We hold a unique position due to our strategic location and the wealth of expertise available to us. And from all over the world, thanks to the hybrid modality these days, online modality particularly. So today we have a distinguished speaker talk to us on next-gen healing designed by algorithms. So Professor Pentiala will explore a simple but powerful idea. What if healing could be designed, not discovered by accident, but engineered deliberately using algorithms? So we are entering an era where intelligent computation is not just analyzing biology, but actively shaping the future of therapeutics and regeneration. So this talk introduces two complementary frontiers that bring us closer to that reality. So today's speaker, Dr. Srinivas Pentyala, is a translational scientist whose work bridges medicine, engineering, and computation. With appointments across multiple disciplines and over 100 peer-reviewed publications, his career reflects a commitment to moving discoveries from bench to bedside. Beyond research, Professor Pentyala has mentored hundreds of students and built programs that cultivate the next generation of biomedical innovators. So the talk has two frontiers, like mentioned earlier. The first frontier of the talk is all about using algorithms to find new peptides. Instead of waiting for things to happen by chance in biology, we use computers to look at genetic information and guess which peptides might be good for the brain and how they develop. Then we check the predictions with special tests to see if they could be useful for treating things like Parkinson's and osteoporosis. The second frontier shifts from molecules to materials. So micro-CT imaging and computational optimization are used to create biomimetic bone scaffolds that mimic the natural trabecular structure. These 3D printed structures are then biologically enhanced to encourage osteoblast growth and functional regeneration, combining engineering precision with biological intelligence. So all this essentially merges engineering precision with biological intelligence. By the way, Dr. Pentyala is a medical doctor, so a lot of the terms I got from Dr. Pentyala, actually. These are not my terms. Um, so the convergence, so these innovations show a real strong connection. Algorithms help us find new molecules. Engineering helps us build structures that can heal. Biology checks if these structures actually work. This isn't just little steps forward, it's a big change to how we design healing systems that think. So now let's see how these technologies are changing medicine for the future. With that brief introduction, over to you, Dr. Pentyala. Please take it away.
Can I share? Can you allow me to share the screen?
Yes, yes, please.
Okay. Just can everyone see my screen?
We are able to see, but Yeah, but it is not in a slideshow mode, I guess, right? So if you click on the slideshow button,
See it now? Yep.
slideshow, yes, we are able to see the slideshow now.
Okay. At the outset, let me thank Dr. Vishnu Penkala for inviting me to this Zoom talk. And for all the friends who are joining us, welcome. I guess it's a good afternoon for you. We are almost in the evening now here in the East Coast. So what I want to talk to you is about how bioinformatics, computational science, and computer science coming together with biologists, with clinicians, and engineers, mechanical engineers. And this is a conglomerate of many fields and involve many people over many years. And what I want to do is give you a brief introduction of our work on this one particular project, which has been going on for the past 14, 15 years. And on that note, I'll be talking mostly about biology, and you might hear some words which you may not even understand, but I'll try to be brief on those complex biological or clinical terms and try to go to the basic work and try to explain how all the computational science and how algorithms helps us to discover new drugs and even actually create artificial organs. And on that note, you can either stop me whenever if you want to ask questions, or we can wait till the end. Okay. So Vishnu, are you able to hear? Is
Yes,
everyone okay?
yes, everything
Okay,
is
good.
good.
Okay. So let me give you a brief introduction about myself. I'm in a clinical department in the Department of Anesthesiology, but I have joint appointments in orthopedics, urology, and other basic science departments also. I've been working at Stony Brook Medical School for the past 32 years. I love doing science and research. At the same time, I love teaching, along with all the other things we do in the hospital in the medical school. Okay. So let me give you a brief primer or an intro to what you'll be getting into actually. A little bit of basic biology. Maybe you heard about these terms, maybe when you're in high school, maybe. So I'm going to give you a brief introduction of these terms, okay? So as you know, we are all made up of proteins. We are um, structurally, functionally living because of our proteins. And for basically, as you can say, proteins are the stuff that you find in meat. So we are all basically made up of proteins. And normally when proteins are made in our cells, they're actually the basic units of a protein is amino acid. And these amino acids, they combine together to form a small chain called a peptide. And these chains keep on elongating to form a complex protein. For example, in the egg, you have albumin, that's a protein. And so many proteins are there in our body, thousands and thousands. So when these proteins are made in our body, they are actually made as a straight line, as you can see here. And basically, there are 21 different types of amino acids can make up any hundreds and thousands of proteins. But basically, there are only 21 essential amino acids. So in the cell, they all make these amino acids come together in different sequence to make the proteins. And these straight chains, they start turning and twisting into secondary structure and tertiary structure, and finally forming the quaternary structure. For example, your hemoglobin in your red blood cell is a combination of four different tertiary structure proteins. They combine together to form a quaternary structure. So the protein has to be in this structure to actually do the function. And that's what we teach in med school or in high school also, how these proteins are made. This is called protein synthesis, involving transcription and translation. So now, when these proteins are formed, look at these quaternary structures. When these are formed, they fold into a nice three-dimensional structure. They're really pretty. Actually, we thought we can actually print them and put it on our ties because we work on many of these proteins. But once the proteins are formed in the cell, they undergo lots of covalent modification. There are different types of reactions called phosphorylation, glycosylation, simulation. They add disulfide bond, acetylation. Lots of chemical reactions happens in the cell. Based on those chemical reactions, this protein performs many number of functions. So that's the basic biology. We are what we are because of the proteins that we make and how they are modified to do different functions. And to actually tell you an interesting thing, in each cell, in our cell, we have billions of cells in our body. In each cell, for every minute, more than billion reactions happens inside our cell. Imagine the scope of this protein biology, because all these reactions, they almost all involve proteins. Okay, so now we talked about how proteins are made, how they are folded, and how they do the function. Let's say once the protein's function is over, what happens to this protein inside the cell? The protein gets digested. That means it is broken down. So the protein, a complex structure, a three-dimensional structure, is now converted into a primary structure. And there are lots of enzymes that work and break down these protein structures. And now they form into small sequences called peptides. And many of these small sequences, again, they are broken down into individual amino acids. And these amino acids, they recycle to form new proteins when there is a need in the cell. That's how protein chemistry happens. So they are made, they do the function, and the function is over. The proteins are broken down into bits of amino acids, again, to be recycled to form functional proteins. So what we realized was when these peptides are made, not all the peptides, peptide is nothing but a small protein. Not all peptides are broken down into amino acids. And this is discovered many, many years ago. And a lot of people work on many of these small peptides. And you'll be surprised to know that nowadays, all our drugs that are being introduced into the market, like your Wegovy, your Mounjaro, your Zepbound, all these GLP-1 peptides, what we use for diabetes as well as weight loss, all those are small peptides. But like I said, normally the peptides are broken down into small amino acids. But these peptides remain in the cell to be remaining as peptides, and they don't get degraded. The reason why those don't get degraded because these peptides, look at these ends. We have one end called the N-terminal end, and there is another end called the C-terminal end, starting from left to right, N-terminal and C-terminal. So these peptides, they don't get degraded to amino acids because these peptides, they undergo a special reaction called amidation. Amidation is a reaction where the C-terminal end is modified with an amino group. Again, like I said, we are going into chemistry from biology. Now we are into biochemistry. So these C-terminal ends of this peptide, a small protein, they're modified and they form and they attach themselves with an amide group at the C-terminus. And this is called amidation of the peptides. Why this amidation is very important? Because amidation actually preserves the peptide from being processed further. That means it cannot be broken down into amino acids. Not only that, this amidation will help the peptide to have some biological activity. Rather than being a small amino acid, the small peptides coming from a big protein now will have its own unique biological activity, doing lots of things in the cell. Like I told you in the beginning, each cell performs more than billion reactions. And imagine to do billion reactions, you need billion proteins or billion enzymes. But we all do it with this small amount of proteins and peptides because they are modified. And this modification of these amides becoming peptide amides will help these peptides to do biological functions. And not only this, amidation is very, very specific. Imagine if you have a big protein, for example, my hand. And now this amidation will only happen when there is a glutamine, an amino acid glutamine on one side and a glycine on the other side. Now look at this small piece on my hand. It has to have glutamine on one side and glycine on one side. then only this can get amidated, or else it won't get amidated. So this is what was discovered in biology many, many years ago. So then people are trying to do basic science research, working on the bench, doing protein chemistries, using different techniques, doing bench research using reactions, test tubes, and all that stuff to identify these glycine and glutamate residues on C-terminal and N-terminal ends. But then what we thought a few years ago was, if we use bioinformatics, instead of doing bench research and chemistry work and the peptides and proteins in a test tube, why can't we use informatics programs to identify these glycine residues at the C terminus and glutamate residue at the N terminus and actually pull out the sequence using software? And that's where the whole story started for us. So here is what we did. So we wrote a one-line algorithm, and we are very proud of this algorithm because when we actually started this project, nobody believed in us. Nobody even thought that algorithms and software can be used to actually derive bioactive peptides. So what we did was we wrote a one-line algorithm, and we have these motifs where we taking the structure of these proteins. Like I told you in the beginning, proteins are complex three-dimensional structures. So we took different motifs, different parts of the proteins, and we wrote the algorithm in such a way that once you put in the algorithm into the database, the algorithm will search for this hydrophobic anchor motif. That's the first site that we want to recognize. Then it goes and finds the site where the protein can be cut, which is called the endopeptidase site. And then we also want the glutamyl residue. I told you in the beginning, we need a glutamine on one side and glycine on one side. So we wanted to make sure that this algorithm searches for a glutamine residue on one side and a glycine residue on the other side. And we actually made the peptide sequence. It can be anywhere between 1 to 26 amino acids in between, or lesser than 1 to 26, or lesser than 26 amino acid. And at the end, we should have this glycyl residue. So on one end, it should have glutamine. On the other one, you have glycine. It can be anywhere between 1 to 26 amino acids. The reason why we chose this number, we can just change the number. It's not a big deal. The reason why we chose this number, because in a machine in our labs, we can only stitch 25 amino acids together. It's very difficult to stitch more than 25 amino acids. It happens in the cell biologically, but in our labs, in our chemistry labs, we cannot stitch more than 25 amino acid or 26 amino acid. So when we put this algorithm and onto the database, we actually used a PAR PSD database where all the structures of these proteins, all the sequences of the proteins are deposited into this database. And it's publicly available, it's free, anyone can access them. And so what we did was we put this one-line algorithm to the database, and it took our computer at that time, which is very slow. It took the computer 2.5 days to actually process the database. And to our surprise, we got 127 matches. Now imagine this database with more than 900,000 entries at that time. Now it has more than 2 million entries in many of the databases. But at the time when we started this project, we have 900,000 entries. And out of those 900,000 entries, our algorithm using these specific motifs to identify those peptides that can be amidated, identified 126 matches. That itself was a great finding at that time. So what we did was we printed all those 127 sequences on a white paper, and we put in some undergrad students to actually search for this sequence. Where are these sequences coming from? And to our surprise, all these sequences, they are coming from, if you look at this on this side, if you can see the pointer, you can see these amino acids with amidation, amide group at the ends. These are all amidated peptide of different lengths. And when we did a blast search, again, another bioinformatics program, where we can put in a sequence of a peptide and ask the program to search for where this small sequence came from. And to our surprise, these sequences came from these sort of proteins. For example, a urinary peptide. This is involved in a disease called lipidodystrophy. Another protein peptide came from a protein called c-MYC protein, which is involved in cancer. And another peptide came from TRH, which is involved in thyroid, your goiter, your endocrine system, your Hashimoto's disease, all these things. And some of the peptides came from fertilization stimulating peptide to mature the egg. And some of them actually even came from HIV protein, actually. So all these tells us that our algorithms became very powerful to screen large databases to highlight these amidated peptide, maybe which will have biological functions. So when this happens, we went a little bit crazy, and we started modifying the algorithm sequence to put in some different amino acids that we like to have, because based upon these amino acids, look at this, these are the 21 amino acids. Based upon these amino acids, whether they are negatively charged or positively charged, there are polar or hydrophobic or hydrophilic, they will be involved in different interactions and doing different functions in the cell. So we can modify the algorithms in many different forms so that we can get many, many hits that can be amidated at the end. So all these are amidated peptides. We choose amidation because amidated peptides are protected from degradation. And not only that, they will have some biological activity. That's what we thought initially. So we started playing with our algorithms. And for each algorithm in a million sequence database, we are getting only 100 hits or 70 hits or 72 hits. That itself is a very, very great finding for us, which make their algorithms very stringent to identify unique biological peptides. Got this, everyone? And so I'm just giving you a flavor of these algorithms. And look at these protein names. All these proteins that we are getting are involved in some disease process in the cell or in the human body. So every one of these proteins are now implicated in some sort of disease. And that's where our algorithms and bioinformatics programs helped us to identify these peptides. So I'll pause here and take any questions. If you have any questions, this is the time to ask so that we can move ahead. Any questions? Dr. Vishnu?
Yeah, I don't see any questions on the chat or.
Okay.
Yeah.
Okay, that's fine. I mean, as you can see, it can be either very simple or become very complex. So I'm going to shift gears.
So Srini, one question, right? On the chat window, Anshu Sharma is asking, what language are these algorithms written in?
Actually, this you'll be surprised to know. This was written in Fortran 4.
Oh, wow. So,
imagine that. I mean, actually, like I said, I did a certificate course in Fortran 4 many, many years ago. And I don't know anything about programming. And of course, nowadays, the language is more complex. We have different things. But for me, this is good enough. Because for me, I need these hits. I don't need any complex things. I mean, imagine from million sequences, we were able to get this small number of close to 100 hits. And that's how this was written initially. And so I don't know if anyone knows about Fortran 4 language now.
Yeah. So this is deterministic, right? The algorithm seems quite deterministic.
Yes.
So I was wondering, PTMs, post-translational modifications, have a high variability, right?
Yes.
So how does your algorithm take care of this variability?
Okay, so what we identified was for post-translational modification, particularly we are focusing on amidation, because I told you amidation protects the peptide from degradation as well as increases its biological activity. For anything to be amidated, you need a glycine and a glutamine and a glycine on the N-terminal and the C-terminal. If you don't have it, it won't get amidated. So we made it very stringent. The algorithm is very stringent. If you look at the algorithm, R is the symbol, okay? R is the symbol for the amino acid that we need. And when you have this R at this end, we can actually increase this. I mean, we can actually make sure that the peptides are amidated. And that's how we made this whole thing very stringent.
Okay, thank you.
So now, okay, we have a bunch of sequences printed on a paper using bioinformatics program. Now comes the fun stuff for us, biologists or people who are in the medical field. Okay, you as a computer scientist, you as a programmer, you as an expert in bioinformatics, give some data on a paper. But will this small sequences, these small peptides, will they really do anything in biology in a cell? So what we thought was we did a very simple experiment because I was trained in what we call signal transduction. So I'll put it very simply. If I slap somebody, they'll respond, right? They'll say, ouch, or they'll slap me back, or they'll cry, or they'll run away, whatever it is. It's the same thing. When a biological cell is slapped with some drug or a signal that comes from outside, the cell responds. And the response normally happens within the cell by increasing a messenger within the cell called calcium. I think everyone heard about calcium. There are two second messengers within the cell. One is calcium and the other one is cyclic AMP. These are the 2 second messengers. So when a cell is slapped in a way or is treated with some drug, some signal, either electrical, chemical, hormonal, whatever it is, the cell responds in a unique way. But the response is inside the cell is resulted by 2 messengers that are generated. One is calcium and the other one is cyclic AMP. And we are sort of experts in measuring calcium within the cell. So what we thought was we'll take a cell and we'll inject this cell with some calcium fluorescent dye. That means the dye within the cell will bind to calcium. And if it goes up, the levels goes up, the fluorescence increases. And then we took the cell under a microscope, or you can do population study in a fluorometer. And then we started adding peptides that we synthesized in our lab. We stitched the peptides together and we started adding these peptides to this cell. And to our surprise, as you can see in the bottom thing, this is the calcium green control. We just have a basic fluorescent straight line. Then some peptides didn't give any response. As you can see, when we add the calcium, no response, no response. See the arrows? It's a sort of a straight line. That means the calcium is not raising up. But this is a amidated peptide. So we were very disappointed when this happened that no response is giving. But remember, we are only measuring calcium. Who knows, maybe this peptide will actually work through cyclic AMP, the second other messenger. But to our surprise again, some of the peptides started giving raise in calcium within the cell. As you can see, the more peptide we add to the cell, the more calcium it raises up. Some gave a response, some did not give a response. So what we did was we again differentiated our peptides into peptides that give a response with calcium, because we are experts in calcium signaling. Maybe we may have to go back to other people to see if they can help us identify cyclic AMP signaling. But as of now, we thought, let's fix on our calcium signaling pathways. So what we did was we took one of the peptide, and this peptide, look at this peptide which is underlined, this one. Look at the peptide again put in red. This is the second one. And look at the peptide that is put in the blue. This is the third peptide. All these peptides were derived from our bioinformatics program. And look at this sequence R at the end. That will tell that it can get amidated. And when we did a blast search in the bioinformatics program, we found that these 3 peptides comes from a protein called GDNF. GDNF is a protein called glial cell derived neuronal factor. And this protein is involved in regulating the brain function so that people won't get Parkinson's. I guess many of you heard about Parkinson's, where when people, they get Parkinson's, they start having tremors, they lose their balance, they cannot walk straight, their facilities, they start working or stop working, their motor functions are lost. So this protein is very, very essential for Parkinson's, to prevent Parkinson's. So when we got these 2, 3 peptides, what we thought was, let's see if these small peptides that we can stitch in a lab, in a machine, if we actually give it to a cell or a biological system, will it do anything? So the first thing what we did was we wanted to see whether it mobilizes calcium. Like I said, whether it's biologically active. And you can see the more peptide we add, the more calcium it releases. And we also found that it is released from inside the cell rather than from coming from outside. So we manipulate these cells in different way. We close the doors of the cell so that nothing comes from outside. So when we add the peptide, everything is released from inside, saying that this is a biologically active peptide doing something inside the cell, generating a second messenger like calcium. Then what we did was we actually grow some neurons. Parkinson's disease is because of the damage of neurons, your brain cells, special cells, that release dopamine. Dopamine is a chemical that actually helps us to stay active. But when these neurons, dopaminergic neurons, they get degenerated, you get Parkinson's. So we grow some neurons, and then we treated these neurons in a petri dish with these peptides. And as you can see, when we add the full length protein, you can see the neuronal connections making more stronger. That means more neurons are coming together to become an active network. In Parkinson's, these neurons lose their connections, and that's why we get Parkinson's. But when we added this full length protein, we get these connections. But at this, yeah, go ahead. Does anyone has a question? Okay. So, but when we also added this peptide, 11 amino acid peptide, you can see you don't need the full protein that is secreted in the brain. You can just inject this a small 11-mer peptide, and you can see the growth of these neurons and connection of these neurons. And we went back to the 5 amino acid peptide. Imagine a 5 small amino acid peptide. And as you can see, the more we increase the dose, the more neuron connections we have. So do we think that this peptide is a treatment for Parkinson's? So we are specialists in cell culture studies and small animal studies. Then we thought that we should collaborate with people who are real experts in Parkinson's. So we approached Udall Center for Parkinson's in Lexington. They are the world's renowned experts in Parkinson's research, using monkeys as their models. So they create a Parkinson monkey, and then when we gave them our peptides, they treated the monkeys and the rats also. They also did experiments on rats, and they showed that all the key factors that can prevent Parkinson's, they are upregulated in the rat. And in the monkey, as you can see, these 90-year-old so-called monkey model, when they are injected with the peptide into their brain, these 90-year-old Parkinson's monkeys, they're running like 16-year-old teenagers. And they did lots of studies with us. We did took the brain out of the monkey, did sectioning, and we showed all the connections reforming with these peptides. And so that's where we stand with this project with the Parkinson's, with the Udall Center in Lexington. They are now applying for clinical trials. But the only problem, the caveat I would say is, you cannot take a pill of these peptides, because peptides, when you eat it, normally they get digested. You need to do lots of chemistry again, and lots of combinatorial chemistry using computer scientists and bioinformatics experts to actually make these peptides non-digestible if you take it as a pill. So as of now, the protocol is to directly inject the peptide into the brain of the patient. And that's what they are planning to do. Yeah, there will be a cure for Parkinson's, but this will be a long road to become successful, because I mean, imagine every patient has to undergo stereotoxic neurosurgery by a team of clinicians to get the cure of Parkinson's. So on this note, when we realized this is going to be a tough project to continue, so we almost gave up on Parkinson's in a way that we told Lexington people, the Udall Center in Lexington, saying that take all our peptides, the GDF peptides, and do whatever you want, because I know this will going to be a long. So this is where we stand with the Parkinson's project, okay? Using bioinformatics derived peptide to actually cure Parkinson's. Then we are looking at our list of peptides. And then we found another interesting sequence. If you look at this sequence, and then we took this sequence and put it into all databases of rabbit, mouse, again, a bioinformatics database where you have all the sequence of all the proteins of different species. And to our surprise, this sequence is highly conserved in every mammalian form, like rabbit, mouse, rat, pig, human. All these are conserved in all animals. And this is amidated peptide. So what we thought was, what is so great about this peptide that is amidated, that is found in all these species when we did a bioinformatics search? So what is this peptide is? This became a part of a calcium regulatory protein called calcitonin receptor. Maybe some of you heard about calcitonin receptor that is involved in making your bones stronger in diseases like osteoporosis. So here is the calcitonin receptor. And to our surprise, when we did basic science, we identified the sequence of this algorithmically derived peptide in the protein that is naturally occurring in the cell. And this end of the cell, which is inside, this end is the inside portion of the calcitonin receptor on the cell. This portion is the same protein the same peptide that our algorithm discovered. And imagine, instead of doing 10 or 20 years of research, trying to find this small sequence in a protein by doing basic science work, you can do it in 2 days. Actually, nowadays, with the computers fast enough, with the programs even more evolved and algorithms more stringent with different languages, you can get these amidated peptides in less than an hour. And but we didn't go back and change the algorithms. We are very comfortable with our old algorithm. And look at this. It took us 10 years almost to work on one peptide. We have more than 120 peptides. And I'm telling many of our friends and colleagues, if you are interested in any of the diseases, take the peptide. We'll give it for free and see if it works in any of your diseases. So when we got this sequence, then we realized, is this really amidated inside the cell? We did bioinformatics to show that it gets amidated using algorithms and scanning the protein databases. But inside the cell, will it get really amidated? And to do that, it will take another 5 years to understand. So what we did was we used a sequest program, a sequest algorithm, which is a publicly available database, very easy. You put in a peptide in that sequest program. And like I said, we don't know what the program is made up of. We just put in our query, put in the sequence of our peptide in the program and say, can it be amidated? And interestingly, it will give you all the steps where this peptide can be modified. It also gives the enzyme where this peptide is worked upon. And then finally tells us that, yes, this initial protein sequence can be amidated by using all these steps, using all these enzymes, and give you a final amidated peptide. We didn't do any chemistry reactions. We didn't work on any enzymes. We just used the sequence algorithm to actually put in the query of your sequence of our protein, and then it will tell you what are the steps it will undergo, including the enzymes that will work on these sequences to give us our amidated peptide. Again, using a bioinformatics program, we actually were able to prove that this can happen at least virtually, but it may happen within the cell too. So that's what we are at. And then we did another program to actually look at the 3-dimensional structure of our protein and see where our peptide is. Normally, for the peptide to be biologically active, it has to be on the outer surface and it has to be hydrophilic. That means it needs to like water to get interacted with other proteins or other reactions. So we actually mapped our peptide to the hydrophilic region of this whole protein using another program. And then went into biology again. So what we thought was this belongs to a calcitonin protein. That means somehow involved with calcium regulation and bone. So what we did was We took some osteoblasts, which are the cells that make bone. We actually grow them in culture in our labs in a petri dish. We took some osteoblasts and we treated this osteoblast with our peptide, which we labeled as calcitonin receptor fragment peptide, CRFP, because it came from calcitonin receptor and it's a fragment, and we call it as a peptide, CRFP. And to our surprise, these bone cells, which are grown in petri dish, started making bone modules in a petri dish. So we are actually able to create bone modules in a petri dish by treating bone cells with our peptide that we discovered with bioinformatics. Then we actually said that will a rat peptide, because there is a subtle change in the amino acid, look at here, between the rat and human, will they work both similarly? And to our surprise, they also work similarly. So both of them can make bone deposits on a bone cell. So you treat a bone cell with this peptide, and the bone cell will now start making bone on a petri dish. Then we thought, what if we take the stem cell? You know, stem cells are undifferentiated cells that actually we have lots of stem cells when you are in a fetal stage, and each of these cells can form or transform into a special tissue. For example, one cell can form a brain tissue, the other cell can form a liver tissue. So what we thought, if this is making bone by bone cells, can we transform a stem cell into a bone cell? So we took some mesenchymal stem cells and then treated these cells with our peptide. And again, to our surprise, within 12 days, a stem cells, which are undifferentiated cells, is now morphed or transformed into a bone cell. So now two things are happening here. Our peptide has the capacity to make a bone cell make more bone, or it can transform a stem cell into a bone cell. And that's where the basic science started going. And now we got very excited. So we thought, does this peptide really exist in a human body? So what we did was, we took this peptide and we made antibodies against this peptide. And I'll tell you why we made the antibodies in a minute. Again, we went to bioinformatics. Again, thanks to you guys, computer scientists, programmers, and bioinformatics specialists. We don't do much of the programming. We use your programs. We just took the peptide and put it into a program called antigenic index plot program. So what this antigenicity index program does is, it will tell you whether this peptide will have high immunogenic reaction. For example, if you took a COVID vaccine, that COVID vaccine will cause reaction to make antibodies in your body. But this COVID vaccine should have high immunogenicity. That means it should have some criteria. If it doesn't have those specific criteria, the vaccine doesn't work. You can put in a vaccine, but if it doesn't have immunogenicity, you cannot make antibodies and you won't be protected. So for us to see whether we can make antibodies, we put in this peptide into this antigenicity plot, and we just press enter, and it gives us an antigenic index plot. And the hydrophilic regions and the hydrophobic regions on the bottom of the line. So anything above 1, that region of the peptide is highly antigenic. So as you can see, our peptide is 90% antigenic. That means we can create antibodies for these peptides. So what we did, go back to again biology. We took a mouse and we injected this peptide into the mouse. And imagine, like your COVID vaccine, you injected a COVID vaccine, you make antibodies. Likewise, the mouse started making antibodies against that peptide. And then we took the spleen of the mouse, we took the B cells, which are special cells that make antibodies, again, going into biology, which is well worked out. And we made factories of cells called hybridomas. So we made factories in a way that we take these cells, we freeze them in our freezers. Whenever we need them, we take the cells out, grow them, and these cells will start spitting out antibodies, which we can use for our studies. So that's what the skill or the method of how hybridomas are generated. Hybridomas are nothing but factories of special cells that you can create for against a specific antigen or a protein that can create your antibodies. So we made these antibodies, and now see what we did, a very interesting thing. While we are writing grants to get funding, of course, all these needs funding, one of the reviewers said, hey, you showed that you can create bone on a cell in a petri dish, and also you can transform a stem cell into a bone cell. And the question that was asked to us is, can you heal fractures using your peptide? So what we designed a study was, we took a rat, of course we anesthetized the rat ethically. We have a animal protocol approved by the committees. And then we actually isolate the femur. If you look at this, you cut the femur, means your thigh bone, you cut the skin, you actually separate the muscle from the skin, and you take the femur and put it underneath a bench press. This is a press, what we call the MTES Quattro testing system. What we do was, imagine this is your femur, the bone. We just drop a heavy weight, a wedge onto the femur. What happens was this femur breaks. And then again, we stitch back the skin, put the muscle back, put the stitches back, revive the animal, and then we injected a peptide, our peptide, into the site where we broke the femur. And to our surprise, after 12 days, we found that new fracture healing started forming quickly. And here is the most critical study for us. And then we stained that structure with our antibodies. I told you we made the antibodies. And those antibodies are detecting the CRFP peptide in vivo in an animal at a fracture site. That tells that the animal is making CRFP in vivo. What we made was in silico using a computer program. But we now proved using our antibodies that the animal is making this peptide to repair the bone during fractures. And not only that, as you can see, this is the fracture site. Normally, when you have a fracture, you actually heal slowly. And this is the slow healing process. But if you add the peptide, look how fast the bone cells are coming together and the fracture is healing. So we showed that now this peptide can heal fractures. Okay? So when we again submitted the papers and when we started writing grants again, another reviewer asked us, hey, you are healing fractures. Can you now heal osteoporosis? And I think everyone knows osteoporosis is the most common metabolic bone disorder where the bone density decreases. And of course, lots of billions of dollars are spent on to cure osteoporosis, okay? And lots of people are affected with osteoporosis. So when a reviewer asked us at the NIH study section, hey, can you also use your peptide to cure osteoporosis? We thought we can do that too, maybe, right? If we get the funding, of course we can do that. So what we did was, we created an osteoporosis model, and it's very simple, but yet very complex. And we took our biology from women. Women who undergo menopause, their bones become brittle because they stop secreting estrogens. Menopause means they don't stop producing eggs, and they stop producing estrogen, and their bones become weak. That's why many menopausal women, they are prone to fractures quickly. And to prevent that, they normally take estrogen replacement therapy or HRT, hormone replacement therapy. Using that model in humans, what we did was we created an animal model. So we took a female rat and we took out the ovaries. So when the ovaries are removed, what we call ovarectomy, the surgery is called ovarectomy, and this animal becomes menopausal. And within 35 days, the animal becomes osteoporotic. That means all the bones of the animal becomes weak. So we took an osteoporotic animal and then we injected the peptide into the animals. And I'm not going to the protocol of how we do it. And then after 35 days, we sacrificed the animal and we took the bone out and we took a histomorphometry. That means to see whether new bone is forming. We use special dyes and we use another software, another bioinformatics software called Huygens co-localization analyzer software, where we can take images of a section of a bone section, put them together to see whether calcium is being formed, because all bones are made up of calcium, and at the same time, new bone is being deposited. So tetracycline is a marker that can identify bone deposition. Calcium is a marker that tells bone formation. That means calcium is there. So when you co-localize this using another software, overlay it, you can see all these two bright sides. They're co-localizing, telling that new bone is forming in this osteoporotic bone. So what we did was then we did micro CT. Actually, we have a small CT machine, CT scanner for small animals, for rats. And we took the femur, the thigh bone of the rat, and we can create images of the cortical bone, that means the outside bone, and the inside bone, the trabecular bone. And again, everything is software written by you guys. We just ask the machine and start pressing buttons, and it will actually separate all the things what we want. It can actually separate all the scans, cortex outer region, as well as the trabecular bone, exactly from the femur. You take a cross section, a transverse section, and you can get all these nice images. We also took the vertebrae and did the same thing. We can take a vertebrae, take a cross section, and actually ask the software in our CT machine to just get the outer cortex and the inner trabecular. Once we got the CT images, then we compared all the animals that we used in our study. As you can see, this is the animal and the femur that got affected when we created the osteoporotic image. We created the osteoporotic animal where the bones become very brittle. Look at the holes that are forming in an osteoporotic bone. The bones become weak because the trabecular connections are lost and the bones become very hollow and weak. But when we treated this animal with this 15 micromolar, very small concentration of our peptide, see how strong the trabecular bone became compared to your vehicle. That means a control injection that we did to the animal. And it started building more trabeculae in the bone. So we are on the verge saying that maybe this will create more bone treating osteoporosis in an animal, which can translate into humans. Okay, we wrote this again, published some papers, and again, we got a query saying that, hey, there is already one drug in the market that can build bone. And imagine osteoporosis, there is no cure, except few years ago, I think 4 years ago, Eli Lilly came out with one drug called Forteo. Forteo is again a peptide. Look at the peptide. It's again a big peptide, around 35 amino acid peptide. Ours is a small peptide, 11 amino acid. To make this peptide, they use biology. To make our peptide, we use bioinformatics. But the question here is, there is already a drug in the market. Is your peptide that you derive from bioinformatics effective compared to this Forteo? And of course, we said we have to try this. And if you give us money, we'll again try an experiment. So we got another rounds of funding. And then we actually did the experiment. I'm going to wind up in a way to make it simple. Look at what we did. We treated one animal with the Forteo, the other animal with the CRFP. We just took 6 animals in each group because it's a complex study. And as you can see, the outer cortex of the bone, this became little thicker when compared to the baseline. But look at our peptide. Look at the thickness of our cortical bone, which is even better than the existing Forteo. Not only that, look at the inside trabecular structure. Look at the osteoporotic bone treated with Forteo. Forteo, Eli Lilly's drug, is really good. Actually, it helps in a little bit of treatment of osteoporosis. But look at our peptide. It is much, much better. So when we reported these findings to NIH, they said, did you ever actually look at Forteo treatment protocol? Forteo can be used only for 2 years. If you use it for more than 2 years, you get osteosarcoma, which is bone cancer. So then the question is, if we treat the humans or animals with our peptide for more than 2 years, will it cause osteosarcoma? We don't know that. So we are now continuing with the studies, Imagine doing these studies for 2 years and 2 months. That's where we are now having our animals in our animal facility, taking care of them nicely to make sure nothing happens to the animals for the next 2 years. To show that our peptide may not cause cancer, but it is much, much better than the existing only one drug in the market, which is Forteo, marketed by Eli Lilly. Actually, Forteo made close to $2 billion in the year that was introduced. Okay? And while this is going on in the study, somebody said that, why don't you create an artificial bone? And on that note, Vishnu, how much time do I have?
You can go as much as you
Okay.
can.
No,
No
I'm going to wind up
worries.
because I'm going into biology. But I mean, imagine the work that you guys do in computer science, informatics, data science, how it is helping us. So while we are talking about this, actually, this idea came from an undergrad student who is working in my lab. And he's working with 3D printers. And he said he got a toy 3D printer and he's showing off in the lab, showing that he has a 3D printer. And then I saw it and I got excited. And I thought, oh, I need a 3D printer in my lab too. And this kid said his 3D printer costed $800 using plastic. Then I said, I think I can actually buy a little bit expensive 3D printer. And the idea is I'm not going to use my own money. I'm going to use your tax dollars, obviously, or grant money. The whole idea is look at this trabecular CT structure. You remember the CT scans we take from the bones, from the animals? What I thought was I can take this CT structure from a DICOM file, convert into a STL file, and apply it into a 3D printer and print a scaffold exactly like what you have in the bone, and then treat this scaffold with stem cells. Remember stem cells, which are undifferentiated cells, and along with our peptide, hoping that on this scaffold, these stem cells, using our peptide, can differentiate it into bone cells. So when we proposed that, and actually we got the funding in the first round of submission itself. So people thought that, yeah, this might help to create a piece of artificial bone. So what we did was we took the CT scans of both femur and the vertebrae. We created a DICOM file in the CT scanner, and we converted into a CAD image by a program called Geomagic. Geomagic, we bought it from Brazil. It's a very, very good program that can convert DICOM files into printable STL files. And then we actually printed trabecular structures using ABS material. They're simple plastic material. When we started this 6 years ago, we never had another type of material that can be implanted into the animal or the human. But I'll tell you what we did. We actually created an artificial scaffold using plastic initially. Then we took the scaffold, we seeded that scaffold into a petri dish, and we treated the scaffold with all sorts of goodies, added stem cells. When I say goodies, all the factors, the serum, the proteins, all the things the cell needs to survive. But along with that, we also added our peptide. And we let the stem cells morph into bone cells. And then after 4 weeks, we took the scaffolds and we just stained the scaffolds with these special stains. I told you earlier, if you stain red with alizarin red, a stain that tells calcium is deposited. And if we have a stain called Wonkosa that will actually stain the osteoid secreted by bone cells, you can see the brown pigment that is secreted by the bone cells. So now these scaffolds, they are accommodating the bone cells. Not only that, they are now making bone on the scaffolds. So then we thought this is very interesting. And then, but the reviewers of one of the papers said, this image is not enough. Show us real cells. So what we did was we took the scaffolds and we stained the scaffolds with nuclear staining. That means every cell has a nucleus. And as you can see these nucleus of different cells, of bone cells growing on an artificial scaffold. And not only that, then they asked, oh, maybe they are just dots. They are dead cells. So we use differential staining to actually show the real cells. And you can appreciate this is what a real cell looks like. actin cytoskeleton within the cell, and we double stained it, and you can see this nucleus. And these cells are growing on the scaffold. These are bone cells, and now they are interacting with each other. And we did a scanning electron microscope to actually show the interaction between these bone cells. And if you scan the surface of the scanning electron microscope with the SEM, you can actually measure the deposits of calcium. And we showed that the calcium is deposited on the scaffold. And then we thought that, is this artificial scaffold as strong as a bone? So we took the scaffold, put it under a testing machine, and which will actually test the elasticity, the plasticity, and the compression strength. So we take the plastic scaffold, the bone scaffold made by bone cells, and then we press it, and we see where it will crack. And we showed that our scaffolds are much stronger than the actual bone. So we can create a bone outside the body and make it even more stronger outside the body. And so then at the same time, MED 610, a special material was introduced, which was approved by FDA that can be implanted into the body. So it got approved to use in dentistry. So now we shifted from plastic to MED 610. Now we created the same thing. We created a scaffold. Now instead of using a petri dish, we thought we should inject into a mouse to see whether these scaffolds can survive within a mouse. So what we did was, we call it as a muzzle pouch model. We actually take a mouse, we isolate the muzzle. We're not taking the bone or anything. We're not breaking the bone. We just implant the scaffold into the mouse to see whether the cells grow on this implanted scaffold once the mouse is running around. So we stitch it back and we let the mouse live for 35 days. And after 35 days, we sacrifice the mouse. And to our surprise, we found that the cells are nicely growing on the scaffolds and depositing bone. And this is a muzzle implant, not a bone implant. This is the test to see whether these implants are rejected by an animal or humans in the later stages, or they can actually accept the implants. When this started showing good results, then we thought we should do a segment defect model using the real bone. So what we did was, we took a femur, the thigh bone again, see what the model is. We cut the piece of this femur out and then put in an artificial bone in. So this is, I'm not going into the technical details of how we actually cut the piece of the femur. We created lots of jigs, lots of mechanical engineer people are involved, lots of PhD students. And then this is how the surgery is done. We actually open up, look at the jig that we put in, and we took a cutter and we cut the piece out of the bone, and we actually put some screws to hold the bone together. And then we stitch back the animal. Look at this. This is the nails, and this is the plate, and this is the break site, as you can see. And then we stitch back the animal and we injected the peptide. And to our surprise, after 35 days, you can see the X-rays and micro CT images. The fractures are nicely healing. You can see now the bone forming in vivo in an animal in a segmental defect mode. And the CT scan also shows that this is the fracture site. And as you can see, the fracture is slowly healing and the bone is becoming stronger again. So when this, and of course, we did the histomorphometry using that special co-localization software to show that calcium is there as well as new bone is formed. And then the surgeons got involved. We had a nice orthopedic surgery group, and they said, hey, can we do it as real implants? So what they do normally during surgery, if there is particularly for cancer, bone cancer surgery, they do pre-surgical learning and teaching because we are a teaching hospital. What they do is they actually simulate the works using 3D imaging. And what they do is they get the CT scan or X-rays from the patient, and they actually manipulate in silico to see whether they can cut out a piece and put the piece back in, having bone implants. And this is what they do normally. And to do this, lots of mechanical tools needs to be devised. So we recruited 2 PhD students from mechanical engineering. And this is what we did. You have a bone. Imagine this is the cancer site. Imagine you have to cut out the cancer. and put the implant back again. So to put the implant, this is the implant we thought we'll put it back again. But to put the implant back in a right position, we need the exact dimension because bones are not flat. They are three-dimensional. Not only that, they have different tissues surrounding this bone. So you need to have the implant right in the region where you cut it out. So what we did was we created jigs, and this is what we did. We created a special jig where you can actually put it in and cut the bone using a saw in these grooves and take the piece out. And to cut the bone also, we put the jig on the bone. And this is where it became a little crazy for us. How do we position the bone, jig on the bone to cut the exact piece of cancer? So we use lots of iPhones and tablets. And this is a nice grant which we got where every one of us can buy a tablet or the most recent iPhone. So we actually put these iPhones and tablets on the focusing onto the camera, and all these 4 tablets or 3 cameras, they image and help us position the jig in a right configuration without any leeway, because the jig cannot be loose. It has to be exactly fit in so that we can cut the piece out and immediately put the artificial bone that we are creating in the lab to put it back onto this bone that we are taking out. Either you take a broken bone out and put a piece in, or take a cancer tissue out of the bone and put your own piece. So the whole idea is now take stem cells from the same patient, because we can take stem cells from fat, take them to the lab, morph them into bone cells on an artificial scaffold, which we created using the X-ray and micro CT of the bone where the patient is having cancer, and then make exactly the same piece that we are cutting it out. Once the surgeons are ready, once the artificial bone is ready, we want to take the piece out and put the artificial bone that we created in the labs. And that is where we stand on this project, going on and on for the past 12 months. So with that, I'm going to stop here. Hopefully we are doing on cadaver bones. That means bones from dead people. We get them from our anatomy labs, and we are practicing, put the jig in and cut the cancer piece out and try to fit in our regular artificially created bone that we make in the lab. So all this happened because of bioinformatics, algorithms. Imagine where we started on a paper, now went into cells to show that the cells are producing bone. Then it went into healing fractures. Then it went into maybe curing osteoporosis. Then we are at the stage of maybe creating artificial bone. All these processes involve computer science, computer scientists, of course, data scientists, and people who are experts in writing algorithms and programs. And without computer scientists and the bioinformatics people, this wouldn't have happened. And on that note, I'm going to end up by saying that I'm glad that this is nicely funded by different funding agencies. We are planning many, many future studies. And of course, a couple of patents also came out on this. And this was licensed to a couple of industries where they are giving some money to actually develop a patch that you can put it onto the skin so that people with osteoporosis can get the peptide as a transdermal patch, like a nicotine patch, so that can get absorbed into the skin and they can cure osteoporosis. So on that note, I'm going to end saying that this cannot be done by one person alone. I'm just the one who talks a lot, I guess, but these are my senior colleagues. Imin Kao is a distinguished professor of mechanical engineering. Michael Hajriguru is the chairman of biomedical engineering. And of course, this is my mugshot. And David is a wonderful bone biologist who is a specialist in animal surgery. And Dr. Fazal Khan is an orthopedic surgeon, particularly on child orthopedic sarcoma, osteosarcoma. He actually treats lots of children with bone cancers. So we all got involved, and these are some of my PhD students and MD students. Some of them now became my colleagues. John is the one who gave us the idea of 3D printing. He played with the 3D printer, so I stole his idea. And Carlos is back in Ecuador. He's now a big shot professor. He started his own company on 3D printing. He did his PhD with me on all these jigs and how to create artificial scaffold. Rebecca, she did a lot of work as a med student. Somehow she didn't like orthopedics. Now she is a neurology fellow in our own medical school. Sai is an orthopedic resident. Sahana is now, she's a psychologist now. She worked on the team as an master's student and an undergrad student. Liz is now a PhD student at Harvard. And Vamik is another PhD student from mechanical engineering who is now working in industry again, working on the same projects of creating artificial. And like I said, without these kids and my good colleagues, this project won't be possible. So I think I'll stop here and I'll take any questions.
Yeah, thank you, Srini. That was really very insightful. Not just me, but others are also saying it on the chat, simply astounding and things like that. But there are also some questions.
Yeah, sure.
So the first question, the first question from Jean Freeman is any type of AI used for the pattern identification? Obviously, there is, right, for the bone structure and things like that.
Yes, particularly for AI now, like I said, we don't have to put it into the database and wait for it to spit out the sequences. You just put in the query, and like I told you in the presentation, proteins get post-translational modified in many different ways. We are only working on amidated, but because we believe these peptides are stable. But for biological functions, other modifications are also very important. And the AI can actually spit out all the peptides that are modified in just like that. And we have very good bioinformatics bioinformatics department, which is gaining a lot of momentum, but they are getting lots of funds to do these studies.
Okay, and so related to that, I was curious if you use off-the-shelf programs, packages to do this segmentation, or you ask your students to do it?
The off-the-shelf programs, in the sense that first algorithms that we wrote, we have it. But anyone can write it. I mean, if you understand the protein chemistry and biology, how they are forming, how they are folding, and how the sites are outside of the protein to do the function, you can actually write your own software. Or you can use the existing outside software that people are now marketing to actually get your, but then you'll get lots of hits. If you don't understand the biology of how these functions, you might get lots of hits. Then you have to go one by one to test biologically whether they are active.
For these image segmentation and things like that.
Yeah.
So do you have students writing the
actually, luckily for us, I mean, our actually, if you look at our computer science and our radiology department, they are big into virtual imaging and segmentation and putting back together. One of the big projects at our medical school is virtual colonoscopy. If you type in, I mean, if anyone, I think most of the audience are young kids, but anyone above 45, they know the pain of getting a colonoscopy. So which is very painful. Now you can do it virtually by imaging getting a virtual imaging, sectioning, and putting them together to see whether there is any problem. And all the faculty in radiology are computer scientists. They are professors in computer science who have joint appointments in radiology. So you can actually create software to do these things.
The reason I was asking is because one of our students, Sarah Peggs, is asking if there is any way that they can get involved with helping in development and manufacturing.
Okay, in development and manufacturing, I would say development, yes, because we are still academics. In academics, we are still a medical school. We can actually collaborate. I can get her in touch with my colleagues in computer science who are really good, who really help us a lot. And she can collaborate with them to do whether she can help them or maybe get some assistance for them for her studies too.
Okay. And there is a question, does it work better with a body than a metal implant?
That's the basic question. The first time when we proposed this, project, that's one thing they ask. Normally, when you have an implant, the implant can be either a steel implant or a titanium implant. Okay, so that's what normally happens. You heard about steel rods, remember? You heard about titanium rods. Now imagine bone. For example, you are 25 years old, you had a fracture or you are a big fracture, you put in a steel rod. Bone is constantly changing around the steel rod. The steel rod remains the same, but the bone is remodeling. Now imagine if you are 40 years old now. Your bones are started becoming weak, but the steel rod remains a steel rod, and then all the bones start fracturing around the steel rod. And that's why all the steel rod and titanium implants, they go for repeated surgery of after 5 to 6 years. Either, particularly in the kids, they are growing, and you cannot put one steel rod hoping that the steel rod will grow too. So that's a major problem with the metal implants. What we are doing is we are taking your own stem cells, we are taking your own and making them into your own bone cells and putting them on your own like a natural scaffold and putting back into your system. So along with your normal bone, your artificial bone that you are putting, we are putting in, it also remodel accordingly based on your biology. So we think these implants are much better than the metal implants.
So another related question seems to be, can you make screws with artificial bones?
Unfortunately, as of now, I mean, these screws are very small. And imagine the screws that we're using for mouse and rats. At least for humans, we have big ones. But the screws that can be reabsorbable screws, what we call, there is material, bone powder, that they fill in when there are fractures. People tried making screws and plates with this bone powder, but they are a little bit soft. They cannot hold the rigid structures. So for many patients, even now, they leave the implant in such a way that the bone filling material will heal and become a bone, but the steel plates and the steel rods or the screws, they remain the same. We don't have anything that can replace the steel plates and screws. We still have to rely on it. But if we create the bone and the bone heals, we can actually, people go in and take the steel plate out and the steel screws out. Some people, if they are in the phase, they'll let it go, nothing happens, they remain there forever. But in some people, they actually go in again and take the steel plates and steel screws. I hope I answered your question.
Yeah, Sarah, the asker, is saying it's interesting.
Yeah, and also on that note, steel, I mean, it's very interesting. In other countries, I heard that steel manufacturing, these plates and steel manufacturing, steel should not rust. And if anyone have bought any steel utensils, if they are really steel, they don't rust. Now imagine what it is, what implication it has when you're putting these steel implants that are not made properly. Infections and all sorts of problems happens. So we try to be aware. A lot of people want their steel plates and screws to be removed after the bone heal.
Okay, and so those are the questions on the chat. There are some. So one of our students, another student, Prashasti, she is also interested in collaborating to contribute to your work. So maybe they can just look you up on internet and email.
I mean, they can drop an email to me, and if they can send me their whatever resume they have, I'll try to, if I can help them, because like I said, I'm not a computer scientist. I'm a biologist. I work in the medical field, but I have lots of my friends, like I said, I'm working here in this same institute for 32 years, created lots of good friendships. We work together and we love to help the students who are trying to come up with all these ideas. And like I said in my talk, many of the ideas comes from my students. I have an open door policy. Either it's a high school student or an MD resident, we listen to them. And of course, they have to listen to me also. So
yeah. In fact, incidentally, this post-translational modification idea that we are working on currently is from a high school student who approached me. So that's how I got into this biology and got interested. So
they're smart kids.
Yeah, they're really. So to close the discussion, I just wanted to ask a question that I myself had. I was just curious, not exactly related to the talk, but so you're also a founder of some biotech companies, right? That's how I remember.
Yeah, yeah.
Yeah. So what do you see as the biggest hurdle in licensing algorithm-driven medical technologies to the broader industry?
Okay. First things, the basic thing. You are a faculty in a university. Okay? So if you are a faculty in a university, whatever you do, whatever you discover or invent belongs to the university. Period. No exception. The university intellectual property office, they'll take your patent, they'll file for the patent. That's a good thing, because filing a patent itself costs close to maybe $50,000 to $100,000. I imagine if we have to spend that money on our own, it's a little difficult as a faculty. So once they file the patent, then they'll get the patent and then they'll put it out to as licensing contract to industry. Sometimes we form our own company and we license the technology from our own university, making a deal with them. The advantage of actually forming your own company and taking your own intellectual property from your own university is you don't have to pay upfront fee. You'll make a deal saying that give it to me, back to me. I'll work in the in the university as well as outside the university, in the company, and whatever comes up, if it makes money, then we'll share. And the university will gladly do that. The second option is sometimes the university says after the patent is filed, they'll ask for industry to come and maybe take this technology. After 2, 3 years, they'll say, oh, nobody is interested. Then we'll say, oh, if nobody is interested, give it back to us. We'll try it on our side. So what we do is we make a deal with them again, because the university will say, just give us a little bit, not more. Just give us a little bit in the future, not even right now. Take it away because nobody is interested in your so-called stupid technology. That's what they say to me sometimes for many of our patents. It's been there for 10 years. Nobody is interested. What do you want to do with this? And of course, the patent life is 21 years. So you need to develop this. So what we do is we take the technology, form another company, or you make friends in the company, give it to them. The problem is now you cannot work on the technology in the university system. If you start working on this technology back in the university says it belongs to us. So you have to be very careful. I mean, walking this tightrope. If you take it out, you should not work it in the university. You need to collaborate with industry or go in the evening or weekends to your own company where you have your people, you hire your people, you raise the money. Then if it works, then you develop it, you go to angel investors, venture capitalists, all that whole thing. And I'm happy that I learned the hard way over the 30 years, formed few companies, some went really big, some collapsed, some technologies are now in clinical trial, partnering with the university, but a couple of them are not partnered with the university. So I went both ways on this round.
Yeah. So thanks a lot, Srini, for all those great insights. I think the audience really liked it.
Thank you. And
And
please send me your feedback. You can look me up. My email is there. Just type in my name. Just drop an email to make because I mean giving a virtual talk on Zoom on a complex topic like this is a little difficult. So I can learn from your feedback. I'm a constant learner. I'm a constant, I mean, perpetual student. Whatever criticism you can give, just give it to me straightforward. I don't feel any bad. Okay, so that I can learn and I can improve my presentation of how to do it on Zoom, maybe. Okay. So
that is very kind.
and thanks, Vishnu. Thanks, everyone. So
So
and
yeah, I just wanted to end with this small token of appreciation from our side.
thank you.
If it was in person, we would have done a little bit differently, but because it's remote.
Okay, hopefully, I'll get it by email.
Yeah, I will also send it by email. Yeah. So yeah, thanks everyone, and thank you particularly Dr. Pentiala for a great session. and we'll keep in touch.
Okay, thanks everyone. Enjoy the weather. Here it's very cold here. So, okay, see
Yeah.
you all sometime soon. Okay, bye.
Yeah, bye. Bye, everyone.